And if at any point anyone has any question, please type it into the chat. So we have two moderators here and fellow instructors. Their names are Mansi Gupta and Enwen Lin. So they are fellow students at Brown University with me, and they're going to be helping the whole division split off part. So just to kind of give an overview of how today's going to go. Today's lesson is going to be very easy, very simple. It's not going to be as difficult as the pre-test that we gave out. So no worries about that there. Essentially what we're going to do is we're going to have a big general lesson, like I kind of mentioned in the email, and we're going to teach for about like 45, 50 minutes and just kind of cover all the categories. And then we're going to break up into smaller groups. So the purpose of smaller groups is to kind of emulate the kind of feel you get in a private school atmosphere where it's more one-on-one and personal. Because I know when there's a big class and it's kind of, then people are more hesitant to ask questions. So when you have a private instructor for you, it'll be easier. So in the form when you all signed up, you indicated if you were a beginner, advanced, or intermediate. So we will be kind of arranging you into groups according to that. And if you want to change your group preference based on how this lesson is going and you think it's a bit advanced and you said you were intermediate and you want to go down to beginner, or if you think it's too easy and you want to go up to intermediate or advanced, just send a message in the chat and we will account for that. But yeah, that should be it. So just a quick like 30-second background on VOTM and JMA. Voices on the Mount, founded by Obed, basically got in touch with me. I'm one of the co-founders of Junior Medical Academy, and I'm a nonprofit organization. And we basically work towards empowering students with medical biology education. And I know a lot of work that Voice on the Mount does is to empower a lot of students in Ghana and also across Africa to have the skill sets that they need to approach the next generation. And that's kind of where this collaboration came from. So we have had responses from students from over 11 countries. Unfortunately, not all of them could be with us today because of Wi-Fi issues. So we are recording this lesson for them so they have access to it and everything. But we're just really grateful to be able to have you all and to be able to learn by your side. And we want to see how just this kind of communal educational lessons transcend not only our state or our country, but our world as a whole. And we can have students from the UK and USA and Ghana learn next to students from Botswana and Nigeria and Kenya. So it's a very cool, very collaborative experience that we're very lucky to have. So before we kind of begin and delve into the lessons, one quick thing that we did want to cover was the JMA ambassadorship. And essentially this is something that we are offering to all the students that complete our course and go through the majority of the lessons, that they will basically be given this international ambassadorship, which is a really big honor. So traditionally, we've only been awarding this to the winners of this International Science Buskers Festival that we're partners with, but we want to extend this opportunity to all of you that are learning these lessons. So the idea is that because we have such intelligent students from all over the world, from not just African nations, but from Europe, from Asia, from America, from North America and South America, we want to give you guys the opportunity to be able to take all these, take what you're learning right now and be able to spread it in your home communities. So I'll give you more information on this both after this session and in later sessions, but this is just a reward that I wanted to make you all aware of. But past that, yeah, I just wanted to cover the outline for today. So outline-wise, today we'll start off by discussing CRISPR, which is a really cool technology that's used for DNA modification. Next, we'll actually switch gears and go to aquaponics. So a lot of you, when you signed up on the form, indicated like an interest in agriculture or farming methods. So this is a more advanced, like scientific farming agriculture method that I wanted to teach you all about, and I think would be very exciting for you all to learn about. And next, we're going to be learning about nanotechnology, which is basically like a way to mobilize miniature robots to go and detect and solve certain kinds of diseases. And lastly, biosensors, which is a way to detect different kinds of disease states. So before I go into the lessons, does anyone have any questions on how today is going to go or what's going to happen today or any of the lessons? You can feel free to unmute yourself or you can type it into the chat. Okay, I think. Okay, cool. Yeah, so I will begin with the lessons if no one has any more questions. And again, feel free to type any questions you have into the chat. And Monsi or Anwen, they'll keep an eye on the chat. So Monsi or Anwen, if you see any questions that I kind of didn't see because I'm in presenter mode, just let me know and I will, I'll make that happen. So without further ado, let's begin with that. Okay, so yeah, today's topic is medical innovations. And first we're gonna study CRISPR. So before we kind of delve into the specifics and the nitty-gritty of what CRISPR is, we're going to start by talking about a story, because that's how I really believe in presenting biology. A lot of it is a story, and this story is like a war that has transcended the beginning of time. Now, we all might see there's a lot of geopolitical conflict on the news. There's lots of conflict in the Middle East. There's conflict between different nations, different groups, whatever. But the biggest and bloodiest war in history is actually fought between bacteria and viruses, with billions, if not trillions, dying in the entirety of the process over the course of history. And the way this conflict works is that essentially just the same way that we can get sick, the same way that we can get infected by a cold or by coronavirus, and we have to worry about that kind of stuff, so can bacteria, and they can be affected by viruses called bacteriophages. So before I kind of go into the more visual aspects of the story, I just kind of wanted to walk you through the origins of CRISPR. So it's like I was mentioning, it's a bacterial immune defense. So just at a higher level, I'm gonna cover this in a lot more detail, but at the higher level, CRISPR in bacteria came in the same way that we form antibodies. So when we get sick, what our body does is that it forms antibodies against a virus or a pathogen, and then stores the antibodies to remember that we had to fight this virus or pathogen. So the next time we encounter this virus, our body can automatically kill the virus, and we don't need to get sick. That's why you can't get sick from the same disease twice in a row, the exact same disease twice in a row. So basically, the way this works, like in this story, is that you have bacteriophages, and these bacteriophages are infecting bacteria. Let me see if I can get my remote control actually to make things easier. Actually, yeah, this is fine. This is fine. Anyway, so we have bacteriophages, and can everyone see my mouse, by the way, or is that not visible on your end? Okay, good. So essentially, yeah, so we had bacteriophages that were, okay, cool. So we had bacteriophages that were infecting bacteria. And as you can see, this big blue pill-looking thing is a bacteria. And essentially, the second a bacteriophage lands on a bacteria, they inject their own DNA into the bacteria. So over here in this diagram, this pink spider-looking thing is a bacteriophage. This whole blue part is just a zoomed-in version of this blue coating on the bacteria. And this is the inside of the bacteria. So the bacteriophage basically injects its DNA into the bacteria, the same way that you can imagine if a spider was biting you and they're injecting their venom into your skin. It's like that, like this bacteriophage is injecting its DNA into the bacteria. And essentially, this is what we call CRISPR. So CRISPR is just basically, it's short for this like palindromic repeat sequence that's within bacteria. So that's basically their own DNA. So as you can see here, this big genetic strain is all at the top is all due to the bacterial DNA. And down here, this big strain here is the viral DNA. So essentially what happens is that the viral DNA is integrated right into the bacterial DNA. So they both become one and the same. And the purpose for this, the reason why this is so useful for the bacteria to do, and you might be wondering, like, why would a bacteria want, kind of going back to this, why would a bacteria want to put the virus's DNA into its own DNA? What's the purpose of doing that? If a virus is supposed to infect you, isn't it dumb to kind of put its DNA into your own DNA? And the answer is actually, it's really intelligent. And the reason it's intelligent is this protein called Cas9. So many of you, if you've heard of CRISPR, you've heard the system been called CRISPR-Cas9. And essentially, what that means is that the Cas9 is a specific kind of protein. So now we have millions of proteins within our body. Some proteins are in the form of antibodies. Some proteins help you digest things. Some proteins help your heart beat properly. These proteins essentially read DNA. And that's a really, really cool facet of what they do. So what they do is that they read the entirety of the viral DNA, as you can see here. Like this pink bubblegum looking thing on top of the DNA sequence is a part of a reading protein that's in bacteria. So as they read the protein, as they read the DNA, as this protein reads the DNA, it basically goes and puts out a message. So the way I want everyone to think about DNA is like a recipe. And we'll cover this actually in our next session. So for the purposes of today, DNA is like a recipe. And what do I mean by that? The same way that if I gave you a recipe for cake and said, hey, add, 3 eggs, add this much flour, add this much baking soda, add this much sugar, and you follow that recipe exactly, that's exactly what DNA is for your body. So they say, add this kind of substance, that kind of substance, this kind of nucleotide. And when you put it together, you can get proteins and other kinds of cool structures and important materials for the body. We'll put it like that. So essentially, this Cas9 protein reads the DNA and is able to interpret it. And this Cas9 protein basically has a sequence that it knows. So the Cas9 protein has a sequence it knows and matches it with every sequence that it comes across. So you can almost think of Cas9 like a very, very well-trained surgeon, that it wants to find the perfect match for the strand of DNA that you give it. So the best, I guess the best analogy for this is that if Cas9 was like a kid, and if you had building blocks or Legos, you have one specific piece of a Lego that's a perfect match for another specific piece of a Lego. So if I gave it this one specific piece of a Lego, it's going to look for it's going to look through all the Legos available, all the nucleotides available, and see when it can match a proper genetic sequence that's a perfect match. So now just kind of getting into the more informative part about CRISPR. So what does CRISPR do? Its entire purpose is to find a very specific sequence of DNA. And like I was saying, yeah, it matches this with the whole Lego analogy. And if it does find a specific sequence of DNA that matches with the sequence of DNA that the CRISPR organism was given, then what it can do is it can edit DNA in 3 different ways. It can add DNA, so it can add DNA into the bacterial sequence. It can subtract DNA, so you can cut out bits of DNA. And lastly, you can even change sequences. So we'll actually get into this when we go into private groups and we look at applications. But one big application of CRISPR in the biotechnology realm is that you can change genetic sequences. So this is huge for cancer, because cancer happens when basically you have a mutation in your genetic sequence, right? So if you can change that mutation, if you can take that error out of there, then that's a huge, huge deal. And that's a big reason why a lot of people in the scientific community is so excited for CRISPR. Yeah, and I know I'm going a little fast right now, so I will slow down just a little, just to pace myself a little more. So before I kind of continue, does anyone have any questions? And again, feel free to speak into the mic, or you can type it into the chat.
Well, no, there is no question.
Okay. Oh, okay. That's a good question, Obed. So CRISPR, I'm actually getting to that in a little more specificity. So right now I've kind of given everyone like a higher level overview of how CRISPR works in bacteria, but I'm going to go a little bit more in depth and show you how it can apply for humans. So that's a good question. So essentially, the way CRISPR works is, I think this analogy sums it up very well. The same, many people may or may not know this, but eagles have very, very good vision. So they have very good vision, so they can spot their prey from afar. So even, I think it's like hundreds and hundreds of meters up, an eagle can spot a tiny mouse hiding within the shrubs. So the same way that in this diagram, this big eagle can go and have vision to see like, oh, what is this tiny mouse in this field of like hundreds of mice? That's exactly what CRISPR does. So in this analogy, the eagle is like CRISPR. The white mouse is like a specific genetic sequence it wants to alter, and all the black mice is like the rest of the DNA. So CRISPR is hyper-specific, and when it finds a specific strand of DNA that it wants to modify, it can zoom in on that, hone in on that, and selectively modify that part of the DNA. So basically another kind of analogy that I want to use to kind of illustrate the point and kind of show you how this works is like, it's kind of like a wanted poster. So the same way that if you have, a population of hundreds of people and then one person commits a crime, then on your wanted ad, you're going to put that one person's face and say, oh, this person is wanted. This person committed a crime. So everyone, let's try to track down this person. Let's try to find who they are. So that's exactly what CRISPR is like. You can think of CRISPR like a wanted ad. So CRISPR, you give it a certain strand of DNA, and then it scans the entirety of the cell to see, well, does this match? So it's like CRISPR is a police officer, and it's given this wanted ad, and it knows that, oh, there's a specific sequence of DNA that I want to track. And now that I know what the wanted ad is, I'm going to look through all the DNA to find what, what kind of DNA am I looking for. So to kind of give you a better visual, I hope everyone can see this, but essentially this is the entirety of the CRISPR sequence, and it has this archive sequence and a normal sequence. So this will keep repeating. So I'm just gonna shut up and let everyone look at one repetition without me interrupting. Okay, so let me just kind of explain what's going on here. So over here, it's like the viral DNA and the bacterial DNA, and CRISPR as a system. So it has, it's like a protein, right? So the protein has this archived DNA sequence. So that's how the CRISPR knows what to look for, because it has this archived DNA sequence. So when it scans DNA, it has this archive sequence, and it wants to match that archive sequence to the other sequence, if that kind of makes sense. Okay, good, good. I'm glad everyone, that's making sense to everyone. So yeah, so this I think this kind of short video kind of sums it up really well, because it shows you how it's like this archive sequence is like a wanted ad, and the overall viral DNA component is like the overall population. So it's just trying to match, well, what matches with this archive DNA, and it's hyper specific. So we'll be getting a little bit more into this in another lesson. I don't want to jump the gun too much, but a big reason why this is so helpful is because in the past, CRISPR, without CRISPR, before we had CRISPR, we were basically using, let's say for cancer, treatments like chemotherapy. And some of you may or may not know what chemotherapy is, but it's like a very generic toxic drug. So it actually kills a lot of your own cells. So it's very toxic to the body. It's basically like modified poison. So a lot of people say that chemotherapy could be good because, you know, it does kill cancer cells, but it's also bad because it kills a lot of healthy cells. So before, when we had diseases like cancer or something, we didn't have a way to specifically deal with it. We would give like generic medication that would harm all cells, that would harm healthy cells and cancer cells. So that's why, if any of you know anyone that's been affected by cancer, you see they get really sick, you see they lose their hair, you see they get frail, they lose immunity. That's actually not because of cancer. Those effects are actually because of the medicine we're giving them. The medicine, makes them lose their hair. The medicine hurts a lot of their organ systems. So it's a really painful process. And CRISPR, with the advent of CRISPR, now we realize that it doesn't have to be painful. Because think about it. If this instead of this viral DNA, if this archive sequence could be changed to say, instead of archive DNA, if it was cancer DNA, then CRISPR would only be looking for cancer DNA. So imagine if this cut was around a cancer DNA sequence, then we could literally cut the cancer out of your genome and you wouldn't have cancer. That's an incredible thing. And that's why there's been a lot of research going into this scientific discovery, because if we can hone this more, if we can modify it and we can control it, then the limitations are endless. Because think about it, this isn't just applicable to cancer or viruses. There's so much more. There are many other genetic diseases, like certain kinds of blindness is genetically passed on because of defects in proteins in the eye. Certain kinds of diseases like Huntington disease or anemia or sickle cell disease. These are like blood type disorders. Those all happen genetically. So it's a very important question of, what if I could literally cut those genetic diseases out of your genome or modify them so they weren't there to begin with? It's a very, very cool process. Yeah, but yeah, it's like a lot of people didn't know that chemotherapy was for the cancer. So it's very cool. And before we kind of go a little bit I think this will be helpful. So this is just another analogy that I'm trying to use to kind of really illustrate the point of how CRISPR works. And I know it might seem a little bit repetitive, but just scientifically, when you have something given to you in 5 different forms, even if it's repetitive, it really helps you to encode and understand. And that's what I really want to do here with these VOTM JMA lessons. I don't want everyone to memorize how this works. I don't want everyone to memorize the names Cas9 or exactly what CRISPR stands for, because I don't care about that. And honestly, neither do you. what we want these classes to do is train you to be the next generation of thinkers, to work in labs, and to innovate solutions when you go to university. And that's why I'm trying to explain things conceptually, so you understand how they work and you can apply these for when you're trying to actually work on scientific discovery. So to kind of give you another analogy, let's say our DNA sequence is this name, like Takaradi Ghana. So if we want, let's say a random mutation happened, and we had the word like Sadza put in there, so it was Takor Sadzadi Ghana, right? That name makes no sense. That's completely nonsensical, and that's then the word is screwed up. So in this analogy, it's like this top word, like Takaradi Ghana, that's our original DNA sequence. That's a healthy DNA sequence. When we introduce the word Sadza in there abruptly, then that interrupts the sequence, and that could actually lead to cancer. So this is actually what cancer kind of looks like, that it's an interruptive sequence that's integrated into your genetic sequence. So when your cells are reading your DNA like a recipe, it's like the recipe is completely screwed up. Like imagine if you're trying to bake a cake, and then it said, add 2 cups of sugar, add 3 cups of flour, add 5 cups of dirt, and then add 4 eggs. Like obviously that would mess up your recipe, because you're not supposed to put dirt in a cake recipe, right? So that would completely mess up the recipe. And that's very much like what happens in your cells. That it's a very, very delicate balance to make the proteins that make you you. Like why is my hair black? Why am I the height that I am? Why is this my skin tone? Why are my eyes spaced this way? Why do I have even saliva in my mouth that can digest food? These are all, like, there's a very fine balance of how your DNA and proteins how your proteins have to interpret your DNA in order to make it all happen. If you interrupt a sequence, even if it's by a letter, even if it's by a single letter, that can lead to disastrous mutations that lead to cancer. And we will be covering this in the next session, the whole cancer DNA genetics. So totally don't worry if it kind of sounds foreign. This, for the purposes of what we're doing right now, I just want you to have a basic understanding. So anyways, what CRISPR would do is it would act as these scissors. So it would go and cut out around, it would go cut out around the word sadza. And the way this would happen is that if we were to go back to this, the archive sequence would say sadza, right? The archive sequence would say sadza. So when the CRISPR protein goes here, it's looking for the word S-A-D-Z-A. So when it's reading it, it would say T-A-K-O-R, that's all good. Then it'd say S, oh, maybe it's a match. A-D-Z-A, that's a perfect match. So it would cut that word out, and then you would have, just a normal word. So this kind of helps illustrate how we could apply this for cancer, right? We could cut out the harmful DNA sequences and potentially lead to life-saving therapies for people. So does anyone have any questions just on that or the way it works? All right, good. And again, always feel free to type anything into the chat, even if I'm moving on. We will get to it, and I'm glad people are liking this so far. I'm glad it's new information. And yeah, this kind of goes to illustrate the point again that you can modify to search for anything, and it's like a very specific search, which I think we just covered. Okay, and this is one more analogy. I know you guys must be getting a little bit tired of the analogies, but just bear with me. So I'll actually be using this analogy a lot later on, both for cloning and for different kinds of cancer therapies. So this analogy will come up a lot, but it'll be a fun analogy. Oh, someone had a question. Oh, but is CRISPR injected into the body, or is it produced by the body? Yeah, that's a really good question. Actually, yeah, thank you. I actually neglected to mention that. So the way CRISPR works is that it was found in bacteria, right? So the way CRISPR originated was, it was like the way we have antibodies, bacteria had CRISPR. So bacteria would use CRISPR to find if they were infected by a bacteriophage to cut out bacteriophage DNA. So what we did, there was someone named Jennifer Doudna. She was actually a professor at UC Berkeley, and one of my friends actually worked under her. So she basically found that, well, this is a protein in bacteria. Why don't we extract the protein from bacteria and modify it outside of bacteria to see if we can use it on humans? So and then she discovered that, yeah, we could actually use it on humans, and we could use CRISPR to go and cut out sequences on any DNA, whether it's baboon DNA, whether it's mice DNA, whether it's human DNA. And that's where we saw how to, modify diseases there. So I hope that understands your question. Awesome, good. And yeah, so let me just kind of delve into the dragon analogy. So this is going to sound very silly and very, I guess, childish for the first maybe like 30 seconds, but just bear with me. I promise it's going to come around. But this was basically, I don't expect anyone to know this, but this is like this is from a cartoon called what was this, Bakugan, where basically you have like it's like a you have like pets, and you can go and battle them. It's like a Pokemon kind of thing. So basically one of the Bakugan were a dragon, and his name was Drago or something, and he basically had a crystal core, an infinity stone that they called it. So based on the color of this infinity stone, over here you can see it's red, so he's a fire type, so he's a red dragon. If it changed, then his type changed as well. So if it became like a white crystal stone, then he became like a light type dragon. If it became a dark stone, he became purple and he became a dark type dragon. If he, you get the point, right? If it's brown, then he becomes a ground type dragon. If it's green, he becomes like a sky or grass type dragon. So basically what the point I'm trying to illustrate here is that when we change the color or the type of crystal that the dragon has, and the dragon as a whole changes, right? So if we change the crystal of the dragon to be red, then he's a fire type. But then if we change it to be white, he's a light type. So likewise, it's very akin to CRISPR, where if I change the kind of DNA that's in the archive sequence, I can change the purpose of CRISPR as a whole. So the same way that if I change from like red to blue, you go from fire type to water type. If I change this DNA from a cancer DNA sequence to an HIV DNA sequence, then all of a sudden my CRISPR goes from being a CRISPR for cancer to a CRISPR for HIV sequences, which is really, really cool. So it basically, goes to illustrate the point that we can modify CRISPR to do basically anything we want, and the possibilities are endless. So right now, what's really cool is that if you guys, I don't know if you know much about the scene of cancer out there, but there are literally thousands of genetic sequences for cancer out there. So if any of you are interested in any of these diseases, be it cancer, HIV, malaria, even COVID-19, and we'll actually cover this a little bit in the advanced private sessions. I might actually cover it now, but we'll see if we have time. But essentially, you can even use CRISPR to detect COVID-19 and cut out COVID-19 from lung cell DNA to stop the spread of the virus. So if any of you are interested, all you have to do is find a genetic sequence or a gene sequence in any kind of cell or any kind of disease that you find interesting. And if you target with CRISPR, then maybe you have like the therapies of tomorrow. Maybe I might be seeing your name on the international news for curing one of these big diseases or finding an end to the COVID pandemic. It's a really cool field. So we'll definitely cover more of this and look at applications of this, so all of you get a better idea of how to use this and how to kind of make and spark scientific change with this. But it'll be really, really exciting. I see there's some questions in the chat. Okay, cool. Yeah, I'm glad everyone's finding this very interesting. But yeah, this is the kind of last part I'm going to talk about before we move on from CRISPR. So a big part about a big part about CRISPR is its ability to modify any kind of DNA, and this has sparked a lot of ethical debate. So we want to encourage you all to not only learn about these from a scientific point of view, but also from all aspects, an interdisciplinary point of view. So we actually might actually have partners, other nonprofits come in later and help facilitate debates and help you think about this all critically. But this is just an ethical question I want to put in everyone's mind. So where's the limit of where we can use CRISPR? So I've shown you all the good applications, right? We get rid of malaria, we get rid of cancer, we get rid of COVID-19, and we can detect these diseases. But what happens when people get too greedy? What happens when I say that, I want my son or daughter to be 6 feet tall. I want them to be tall. And what happens if I find a gene that's responsible for your height? And there are many genes that are responsible for your height. But what happens if I find a few and edit a few? So is it ethical for me to modify the height of my child? What if I make them 7 feet instead of 6 feet, right? What if I say that, I really like blue eyes, so I want them to only have blue eyes. So I'm only going to express genes that allow them to have blue eyes. And what happens when that becomes a thing? If CRISPR is widely available, then a lot of you might be thinking, okay, well, that's not a bad thing, it's just height or whatever, sure. But you have to think about the socioeconomic issues there too, which means that if CRISPR can only be afforded by rich people, then what happens if I can pay $10,000 and guarantee that my child's IQ is twice what it should be and guarantee my child's intelligent? Then isn't that unfair that rich people can pay for their children to be genetically perfect and never get sick and be the smartest and outcompete everyone that isn't able to afford those kind of therapies? So that's definitely something that we need to consider, and definitely something we need to be mindful of. And there's a lot of debate about this, that, you know, what if there's an error in the CRISPR mutation? And what if you have babies that are born with 3 legs instead of 1? What if you have babies that are born blind because, you know, you forgot to, in trying to express blue eyes, you down regulated the gene that's responsible for encoding and visualizing the environment? Then that's also an issue, because then once that child is born, you know, it's unethical to kill that child. So you might ruin their life, and you might give them life-altering diseases that that you can't quite take back. And that's definitely something we need to be mindful of. So kind of, so yeah, this would be kind of a good place to take any questions before we move on to the next topic. So does anyone have any questions? Just feel free to put in the chat. I'll wait for like 10 seconds before I move on. Interesting. Okay. And just for when you go into your private groups, so I'm saying this to Mansi and Anwen, who will be responsible for the beginner and intermediate divisions, have a discussion with everyone about what they think about the ability to genetically modify DNA and the whole implication for designer babies. That is it fair for us to design babies perfectly the way we want them, so they have the exact same gender that we want, the exact attributes that we want, the intelligence that we want? Is that okay, or is it unethical? Definitely we should have that discussion. Can the CRISPR procreate? That's a very interesting question. So no, it cannot. So CRISPR is just a protein. So it can't procreate any more than the saliva in my mouth can procreate. So it can essentially cut out DNA, modify DNA, or add in DNA. It's basically like a robot. So it can't procreate, or it can't create babies on its own. So for that procreation process, you still need like a mother and a father and everything. Where CRISPR comes in is after the egg cell and the sperm cell unite and the baby is developing in the mother's womb, the question is, well, can I inject CRISPR proteins into them to modify their DNA as they're developing, to make sure that their heart is perfect, or to make sure their height or skin color, whatever, is perfect? And you know, that's basically how the process would work there. But these are really good questions. Continue to ask questions in the chat, and if I miss them, I will go back to them. So it's never too late to ask a question. All right, and this is just some kind of, these are just some critical thinking guideline questions, but just in the light of the fact that CRISPR lets us add, subtract, and modify DNA, I just kind of want everyone to think about diseases that we could cure with CRISPR and how we could apply it, and what kind, and how we could think about modification experiments. So does anyone, like, can everyone just type like one kind of disease into the chat that they'd be interested in seeing if CRISPR could work for? Like, just do it offhandedly, nothing too advanced or anything, but type in a disease, be it like cancer, blood diseases, Huntington's disease. Just like type something into the chat, and I'm kind of interested to see what you all think that we could do with CRISPR. Malaria, definitely. Yeah, that's an interesting one. Diabetes, that's very interesting, because diabetes. So the thing with malaria is that malaria is more of a bacteria, I believe, that well, it's kind of like a bacteria, like a protozoan, but we don't need to go into that. But it's a foreign organism that goes and attacks your body. So maybe if there's some way for CRISPR to modify our immune cells such that we recognize malaria more quickly, or if we can recognize, if we can strengthen our immune system in a way to be resistant to malaria, that'd be very interesting. For things like diabetes and stroke, those can actually be passed down genetically. So it's the whole nature versus nurture question, that if your parents had heart disease, if your parents had diabetes, you're also more likely to have heart disease and diabetes. So that obviously means that there's a place in the genome for that diabetes or heart disease to be located. So can we excise that fragment of DNA out that's responsible for diabetes or stroke? Influenza is a really interesting one as well. So we'll actually cover this in the applications for COVID with applications for CRISPR with COVID. But when viruses replicate, they actually have to give out their own DNA, right? The same way that the bacteriophage injected its DNA into the bacteria. When the influenza or COVID virus infects us, they also inject their DNA into our cells. So if we can use CRISPR specific to their DNA strands, then we would be able to basically stop influenza or stop COVID from replicating and make ourselves immune to COVID. Because if we could eliminate their DNA before it even reaches our cells or before it can even infect us, then, who cares if you have COVID if you're not, if you're not getting sick from it, if their DNA isn't hurting you or killing your cells. What a disease that can affect your growth? Yeah, so there are certain diseases like Huntington's or chromosomal abnormalities. So if you have trisomy 21, that basically means that if you have an extra X chromosome, these happen when babies are forming. That can shorten your lifespan, that can shorten your height. that can also lead to heart defects and everything. So if we can go and use CRISPR to fix that and not have that extra X chromosome, that could be amazing for people. And can CRISPR ever make a mistake when modifying the DNA? Yes. This is a very, very good question. So this is why CRISPR is not as widely used right now, because it's definitely error prone. So a lot of when a lot of people look at cool therapies and cool kinds of scientific innovations like CRISPR, they think, well, obviously it would work, right? Theoretically, it sounds perfect. But when you do things practically, there's always an error rate. So there's always some risk of CRISPR malfunctioning, of editing the wrong sequence, of accidentally putting in extra DNA, and it could cause more harm than it was put there to modify. So that's why a lot of people are saying that we need more research into CRISPR. Sickle cells, absolutely. Sickle cells actually happen because of a mutation in your genetic sequence. So I think it's an extra amino acid. I don't want to say the wrong amino acid, but essentially, it's basically an extra. It's an extra modification. It's a mutation of a certain protein. So if you modify that, we could absolutely prevent that from happening. So that's a very good question. So yeah, I'm kind of glad we got a lot of talking about that. So we're actually going to shift gears drastically now and go into something more agriculture related. So this is a very interesting intersection of chemical engineering and biotechnology for agriculture. And I think for a lot of our students, even the ones that have to see this through a recording, that are at home, that live in more rural regions where farming is a lot more prevalent, this might be a very, very interesting system for you. So aquaponics is it's not very well known, but it's a very, very cool system. So it's a combination of aquaculture and hydroponics. So aquaculture is basically growing, it's basically like fish, like having fish and keeping fish as pets basically. So that's the bottom part. That's aquaculture. The top part is hydroponics. Hydroponics is growing plants on pebbles and having the water kind of seep through their roots. So normally when I'm growing plants in soil, that restricts some of the oxygen that goes to the plants and restricts some of the sunlight because the soil like covers it all. But if I grew plants in pebbles, then that allows for more oxygen to reach the plant roots, which is good for the plants, and it allows for the water to kind of continuously be drained through the roots. So the way the system connects the two is that if everyone sees the fish at the bottom, so the fish water that they're excreting ammonia in, their waste is very nutritious. So their waste is actually pumped up to these plants. And these plants are on pebbles, remember. So they're not on soil or anything, nothing dirty. They're on clean pebbles. So the water actually seeps through the plants, and all of it goes through the pebbles and everything. And the plants absorb all the nutrients in the water. So all the fish waste ammonia that would normally be toxic for the fish, they absorb all that to grow. And then that filters out the water, and the clean water trickles down to the fish as a result. So it's a very interesting system where you have fish waste that goes to the plants. It's natural fertilizer. They absorb it to grow, and you have clean water coming down. So it's good for the plants because they get nutritious waste, and it's good for the fish because they get clean water. It's a really self-sustaining system. So, and over here on the right is what this looks like. So at the bottom, it's this big fish tank. And there's a pump that pushes the water up to the plants, and the plants absorb all the nutrients, and then they drain down from the pebble bed and go down a drain pipe down to the fish. So this is, if any of you are interested in, I guess, more material sciences or more hands-on work, this would be very interesting for you. So essentially, I actually did this experiment when I was it was like half a decade ago, very, very long time ago. But when I was in I think 7th grade, I did this experiment at home, and it was very, very successful. So essentially, I just wanted to show you what a real, like not just a diagram, but what a real system looks like, which is why I have these images. So down here, if everyone can see, I outlined it. It's the tilapia. So this was a tilapia. It was a rescue fish that I got from and basically what it was doing was I was feeding it food, and it would excrete waste, right? So it would have waste that's in the water. Then over here, if you see this is the pump. So it would absorb all the water and it would send it up. And over here, it would come out like a fountain. So it would come out like a fountain onto the plants, and the plants over here would be growing. So these were seeds that I planted. And then the water would seep down through the pebbles, and it would come down through this drain pipe and it would come back down to the fish. So it's dirty water that goes up, clean water that comes down. It's a very, very cool system. So the way it works on a chemical level is really, really interesting. So the fish waste is chemically ammonia. So that's NH4. So what happens is that the ammonia, when it goes up, is brought to the plants, and it's brought to the pebble bed. And on the pebble bed, you have naturally occurring bacteria called nitrifying bacteria. So all they do is they actually convert the ammonia from ammonia to nitrites and nitrites to nitrates. So ammonia is NH4, then it converts it to nitrites, which is NO2, and then it converts it to nitrate, which is NO3. So essentially, it basically just cleaves the hydrogen off because of the presence of water, and then it adds oxygen from the environment. So it's a very organic, very self-sustaining process. And the cool thing is that plants can't naturally absorb ammonia as well, but nitrates are amazing for plants. So the second this conversion happens, the plants absorb the nitrates up really well, and then they begin to grow. And then again, the nitrates can be toxic for fish. If anyone has ever had a fish, you know that, if you leave the fish in its own tank and you never change the water, then it's just, eating and excreting waste in the very water that it's living in. So if it's breathing in water that's waste, it's going to die because that's toxic for the fish. But what's toxic for the fish is really good for the plants. It's a very cool system. So just to kind of give you a rundown of how this chemical cycle occurs one more time. So just one more time. Essentially what happens is you feed the fish, right? The food goes in the fish and the fish, poops it out as ammonium waste. The ammonia waste gets modified by these nitrifying bacteria. They're called nitrosomonas, nitrospira bacteria. You don't need to worry about that. But essentially it gets converted to nitrites and then nitrates. And the plants take up nitrates and they use it to grow. So this is a more visual way to look at that. So before I proceed, does anyone have any questions on this chemical cycle? Or does anyone have any questions on how this works or why this works? Because I know it's not a common system at all, but I just want to make sure everyone understands it. Okay, looks like it's good, but again, feel free to type anything to the chat. And again, you can always unmute yourself and then say something if you need to. And again, the way it works, as I was explaining, is like the plants are a natural biofilter and the clean water seeps through the pebbles. And yeah, it helps the fish to stay healthy. So this is just a repetition of what I was saying. So just to kind of show you the results of the system. So when I did this experiment, on the left, you can see that these were plants grown in soil. And I used the premium kind of fertilizer. I used the best kind of fertilizer on the market, like Miracle Grow or something. It's one of the best fertilizers on the market. And on the right, I have my aquaponics plants. So you can see that the aquaponics plants, they're over twice the size of these plants on the left, right? They were actually taller than me. So they were very, very tall. And their leaves were huge and they grew really well and they grew really healthily. And a big reason for this is that they used organic, they used like organic waste products and they grew really tall, and that's a really good thing. And someone asked in the chat, let me see. Someone asked if autism could be cured by CRISPR. I didn't get the answer. Oh yeah, thank you for taking me that. That's a really good question. So autism there's several different factors to autism, but some of it has been found to be genetically related. So there are certain genes, I don't want to misquote them, but there are certain genes, I've had friends that have worked on this research at UCSF, but basically there are certain kinds of, I think it starts with a P, but I'm not going to misremember. But there are certain genes that are in their genetic sequence that actually lead to deficiencies in mirror neurons. So autism is a disease, not a disease, but it's a neurological kind of condition where there is difficulty in interacting with others and socializing, but also in relating to others, in empathy. So parts of the brain that should have excess of mirror neurons, which allows me to feel bad for you, or which allows me to empathize with you or interact with you, socialize, there's a deficiency there. So that's partially related to genetics. So one cool thing CRISPR can do is that it can downregulate the parts of the genome that are responsible for autism and the deficiency in mirror neurons, which can help to alleviate some of its effects. Can be used to, yes, exactly, Obed. CRISPR can be used to cure a genetic related disease. So it only operates on the level of like DNA, but it can also operate in the level of like proteins and RNA, and we'll get into that in the advanced private group chats, in the advanced breakout rooms. But yeah, and that basically is it for aquaponics. It's pretty short, but does anyone have any questions on aquaponics? Mental disorders. So yes, it's possible for mental disorders as long as they have a root in genetics. So CRISPR isn't like a cure-all medicine. So if I'm feeling sad, I can't take CRISPR and make me happy. But what we've been starting to see is that conditions like depression have been genetically rooted. So there have been scientific studies that show that, well, if your parents suffered from depression, you're more likely to suffer from depression. Or if your parents suffered from suicidal tendencies, you're more likely to suffer from suicidal tendencies. So there is a genetic link that's been found. It's just that it's not always that black and white that this gene is responsible for this disease. Sometimes it's a collection of 50 different genes that's responsible for a certain disease, and each of the genes have different kinds of functions and everything. So it's a little more complicated than, oh, CRISPR can be used to solve mental health diseases, but technically, in theory, if you can isolate it to a certain kind of gene that's only responsible for that mental health condition, then you could technically alleviate some of its effects. So that's definitely a good point. And Miriam, can our body reject CRISPR as it came from bacteria? So yeah, so that's a really good and really important question. So that's a lot of the modification work that scientists have to do, so that CRISPR is accepted by our bodies and it's not attacked by our foreign body cells. So the way you do this is that you might wonder, how do viruses get past our immune system? So they actually have a coat around them that has proteins that's expressed on all of our normal body cells. I won't go too much into the immunology component at this moment, but one way to modify CRISPR is to do that, to put a coat on it around the protein that makes the body think that this is one of the body's own protein. So that's a very good question, but that's how we can adapt it so it's healthy for people to use. So that seems good. I'm going to move on, but really good questions. Feel free to keep asking questions, by the way, as we go along. We'll just skip over these really quick, and we'll because we're a bit short on time, and we'll go to nanotechnology. So nanotechnology might be interesting for a lot of you. So nanotechnology, fundamentally, you can think of it like miniature robots. They're really, really small. And to illustrate how small they are, if I took one strand of my hair, think about how thin, how small one strand of my hair is. 5,000 of these tiny, modifying robots can fit on one strand of hair. So these are really, really tiny. So to these kind of, nanobots, your blood cell seems like a mountain. It's just one tiny blood cell is like massive to them. So it's a really cool kind of developing area of science. So essentially, these have multiple functions. So one function they serve is to target specific cell proteins. So this is like CRISPR, but on the protein level, where the same way that I said that CRISPR can target cells because of the DNA that they have, these kind of nanotechnology, we can use nanotechnology to target certain surface proteins. And the way that I like to illustrate this to people, and we'll actually cover this in our biomarkers unit next week, but the way that this works is that the same way that everyone has like hair on top of their heads, right? And everyone has different hair. Like I might have like big, messy, thick hair. If Monsi might have like longer hair. Someone might have curly hair. Someone might be blonde. Someone might have short hair. It all varies, right? Everyone has different kinds of hair. So like that, you can think of proteins on the surface of cells as hair, right? They're different. Every cell has a different kind of protein, and it indicates what kind of cell they are. So a blood cell will have different proteins on top of it than my lung cell, which will have different proteins on top of it than a cancer cell. So we can use these kind of nanotechnology and these nanobots to target specific kinds of proteins and eliminate those cells or modify those cells. So in this example, I'm giving you the example of this is an example of a nanotechnology device, and this is an example of a cancer cell. So cancer cells have long been known to express different kinds of modified surface proteins than their somatic variants. And what that means is somatic is another word for healthy body cell. So if I have a normal lung cell, it'll be healthy and it would have a normal protein on it. But if I have a cancer lung cell, a cancerous lung cell, then it would have a very weird protein on top of it. And that's very easily identified by one of these kinds of robots. So it might be a little interesting or difficult to wrap your mind around, because whenever we think about robots, we think about these like big, these big kind of machines. But I want to illustrate the point that these can happen on the microcosm level too. So does anyone have any questions about this before we kind of talk more about this process? Okay, yeah, I'm glad everyone's getting this. So again, this goes with the whole eagle and mouse analogy, that nanotechnology targeting is very similar to like an eagle trying to go and target a mouse and single it out. The same way that we want to single out like cancer cells, for example, from normal healthy cells, because we don't want to kill normal healthy cells. And that's a bit of how this targeting mechanism works like. So what's nice is that we don't just have to kill cells with nanotechnology. We can tag cells, we can deliver drugs, and we can selectively target cells and kill cells like I was talking about earlier. So let's walk through each of these slowly. So what do I mean when I say tagging cells? We can say this like if anyone has pets, like if anyone has a pet dog, then you probably have a collar around your dog to tag your dog that say that, oh, this isn't a stray dog, this is my dog, this is my pet, and this specific collar means that he's my pet. So whenever someone on the street sees that dog, they can say, oh yeah, that collar that collar means that the dog belongs to someone and it has their information on the chain or whatever, and I can return the dog to them. So you're kind of flagging it. Or if another analogy might be that if I'm grocery shopping and I'm shopping for apples, and I see all the apples are healthy, they're normal, they're ripe, and one apple is rotten. So the way it would be tagged is that, that apple is brown, it's like wrinkly, it gives off a bad odor. So that's a way of like tagging it. So if I just showed you the entirety of all the apples available, you'd be able to say, oh, that apple is rotten because it looks brown and all that, and it's visually tagged for you. So now when we're talking about nanotechnology, obviously these things don't have eyes, right? These are robots. So we're training them to tag certain kinds of cells by adding on specific kinds of proteins to them. So you could think about it like a factory quality inspection manager, where if you're looking at a lot of your product, if you have, like a rotten product or a broken product, you kind of tag it and say, oh, this is defective, this is dangerous. So a lot of people have been using nanotechnology to go and flag certain kinds of cells that are cancerous, but the body hasn't caught. So some of you may not know this, but in any given second, there's thousands of cancer cells in your body. And what I mean by that is that there are always mutations happening in your body. There are always DNA replications going wrong. There are always cells that are harmful for you in your body. But the reason why you don't have cancer is because your body is able to see these mutations, and it's able to rectify them and basically cut them out immediately. But sometimes the body misses that, and the body doesn't realize that a cell that divided improperly is improper, right? Sometimes a quality inspection manager might screw up, and you might have like a broken product that comes to your house, or you might have like a rotten apple that lands up in the grocery store because someone just missed it. So that's how cancer happens. That your body's immune system missed this bad mutated cell, and now it's going to grow into a cancerous tumor. So where nanotechnology can come in is that they can screen more rigorously, and they can say that, oh, this cell looks like it's mutated, and this cell has a weird surface protein. So then they flag the cell and put a specific kind of protein that the body knows is bad on the cell, and then the body just kind of destroys that cell. So does that make sense to everybody? Got you. Okay, good. And also a cool thing about nanotechnology is that you can deliver drugs selectively. So let's take us back to the chemotherapy example. And I use cancer as an example a lot because it's one of the leading diseases in the world, like one of the top 10 for sure. And it's a very interesting genetic disease because there's so much work being done into it, yet there's so little understood about the different forms of cancer and malignancies. And I suspect a lot of you, from what you put in your responses, that a lot of you are interested in cancer, have personal connections. So that's why I'm using it as an example a lot. But let's go back to the example of chemotherapy. So when we're trying to deliver chemotherapy to a patient, and a patient takes their chemotherapy medication, what happens, right? The chemotherapy attacks all the cells in their body. So the chemotherapy, let's say I have lung cancer, it'll affect my healthy cells as well as my unhealthy cells that are cancerous. So that's why I might have lots of lung pain, I might throw up a lot, I might lose my hair. So that's a very bad thing. But let's say that I could selectively deliver chemotherapy to only the cancer cells, right? So instead of you having to take, the way it works right now is either through injections like IV intravenous, they put it into your bloodstream, or you take a pill for chemotherapy. And that goes everywhere, all throughout your blood, everywhere in your body. What if I had nanotechnology, these miniature robots that were given a chemotherapy molecule each? So a very tiny dose. So let's say tens of thousands of these robots, and each was given a very tiny dose, a few molecules of the chemotherapy drug, and then I put them into your bloodstream. And these robots were specifically attuned and specifically programmed to only attack cancer cells, to only go and bind and find the protein receptors on cancer cells. So then they would only attack, let's see if I can find a picture for this. Yes, this is perfect. So they would only go and only attach themselves to a cancer cell, and they would inject the drug or whatever into the cancer cell. This is an example. This picture is an example of tagging instead of drug delivery. Okay, this is drug delivery. So yeah. So again, this would be selectively targeting like these kind of mutated proteins on the cancer cell and only putting in that much of the drug into the cell. So you're taking the therapy and making it on a very individual cell basis. So that makes the therapies that much more attuned, that much more precise, and that much more almost harder to conduct. So there's also some downsides to it as well. And this is a whole tagging cell process where this is a bit more glorified and a bit more colored and everything to give you all a better example of how this works. But this is a blood cell. And let's say I wanted to tag the blood cell for some reason because I saw some harmful proteins emerging on it, and I think that it might be cancerous. So it would put, like, obviously I put light here to illustrate the point that it's tagging. It's not actually light. It would put in a specific kind of protein that the body knows is bad. So the body would know to kill this kind of cell. So that's that. And we already covered selective targeting. So that should be good. So before we move on, any questions on these before we go on to more applications of nanotechnology? Okay, good. So yeah, and this might be interesting for a lot of people, but here is a type of nanotechnology called biohybrids. So in the chat, are these solutions in use or are they still in the process of being developed? That's a good question. So the solutions, it's kind of both. So some are in clinical trials in like mice to see if we can make them effective, we can make them safe and compatible with the body. And some are still in the testing phase because these things, because of their technologic nature, what happens if there's an electrical current in the body, or what happens if they malfunction and they're still in your bloodstream and they could potentially damage healthy cells. So there's a lot of health and safety questions, but that's a very solid question. Yeah. And there's a whole rise of biohybrids. So what biohybrids are essentially they're like natural algae cells. So algae are these biologically occurring organisms. They're single-celled organisms. If you've ever been to a beach, you've probably seen a lot of them. So they're coated with magnetic particles. So what happens is that I take some of these algae strands, and they're very simple. They have very simple genetic composition. They have a very simple cellular structure, and I coat them with miniature magnetic particles. So then if I put this into your body, then I can guide these algae particles using magnetic pulses in the body. So magnets shouldn't affect your bloodstream too much. So if I put very weak magnetic currents in your body, then I can guide where these particles go. And what's interesting about algae is that they basically have a property that they release anti-cancerous molecules as they kind of degrade and go through your bloodstream. So I don't know if any of you have heard, if you like one really good thing for you, if you have a lot of vegetables, lots of greens like spinach, broccoli, that kind of stuff, that's all anti-carcinogenic. And what that means is that they release particles that have been known to kill cancer cells and help to prevent cancer and help regulate the body. So that's why a lot of people say, eat your vegetables, eat your greens, because they're good at preventing cancer. So one effective way of doing this, instead of eating greens and stuff, is putting these biohybrids in your body, putting this algae coated with magnetic particles in your body, putting in magnetic pulses so they travel throughout your body, travel to areas where you have cancer. Let's say I had pancreatic cancer. So if I put in magnetic pulses to guide these biohybrids to my pancreas, then as they degrade to the process of reaching my pancreas, they would release a lot of very good enzymes and very good particles that would be able to combat and degrade the cancer cells. So it's a very kind of cool process. And yeah, I know there's definitely more complexity to it, but we will actually get into that over here too and cover some of the other applications, not just biologic. So one thing we can do is that we can modify nanotech, any kinds of nanotechnology devices that we have, be it algae, be it a biohybrid, and we can make them activated by light. So we'll actually be covering this in a later neuroengineering unit, I believe, that we have. But essentially, what we can do is that we can modify these kind of proteins to only respond to a certain wavelength of light. So everyone knows, if I put on like blue light and I shine it on my skin, nothing happens, right? My skin is okay with having like light put on it. If I put in like a red colored light or something on my skin, that's perfectly okay. But because the light happens at a certain wavelength. So if I could modify a specific kind of protein onto these nanotechnology devices such that they respond to a certain wavelength of light, like a blue light or like a red colored light. Then every time I shine that light, then these kinds of nanotechnology devices would be activated and they would do whatever function that they have. And it would be good because this is a neutral stimulus, right? Like shining on a colored light onto my body doesn't affect me in any way. It doesn't hurt my cells. It doesn't give me diseases, but it's a good way of signaling to biohybrids or nanotechnology devices that we should activate and we should go and take a step towards, fighting cancer or clearing up the bloodstream. Some people have used nanotechnology to help clean up their arteries and to make sure that they don't have like heart disease and or they have like, or to clean up like calcification plaques within their heart to make sure that they don't get a heart attack. There's really cool applications. Does anyone have any questions, by the way? I know Ato, do you have any questions? Okay. How do you create these magnetic pulses in your body? So this is administered externally. So scientists would get these kind of magnetic devices. Like if you've ever had an MRI, that in itself has like very big magnetic field waves. So you can have similar devices which have like light magnetic pulses that pulse throughout your body and guide these particles to certain parts of your body. And even other cool things we can do with nanotechnology. Obviously, you can repair damaged tissues. So if for people with deficiencies in their body where you lack white blood cells and fibrinogen and fibrin, that's basically, and platelets work together to heal a wound. So let's say I accidentally cut myself with a knife and I had a gash here. Then my platelets would be basically going to the gash to cover it up so that I don't lose my blood and to cover it up with some kind of seal. And that's why you get like scabs, because your blood cells kind of, your white blood cells and platelets make those scabs so you don't lose your blood. So essentially, what if you have a defect in that and you can't scab and you keep on losing blood, you could technically use nanotechnology to go and repair these kinds of damaged tissues and put in miniature microfibers to go and patch up these wounds so you don't lose a lot of blood. So there's a lot of interesting work being done there. And even past that, there's really cool work being done to detect toxins and take health measurements. So a lot of companies, specifically in the Bay Area, are working with this thing called electronic skin, e-skin, where basically I can put on this kind of, as you see here, I can put on this kind of layer on my hand, and it basically merges. Sorry? Okay. So basically, I can put on this layer on my hand, and what happens as a result is that it can track certain kinds of protein expressions on my hand. So some people are trying to do this with heart disease. So an interesting question can be asked that, can I predict that you're going to have a stroke or a heart attack before you have like a stroke or heart attack? And a lot of people might say, well, how do you do that? A heart attack happens because of a spike in cortisol within your body. And cortisol is like a stress hormone. And when you have high levels of cortisol in your body, combined with other kinds of defects in the heart, and we cover that in our heart unit later on, when you have this cortisol buildup, if you had like this kind of miniature nanotechnology e-skin, electronic skin, then they could detect if you have an increase in cortisol proteins in the body. And the second it detects that spike in cortisol proteins, it can immediately notify health authorities or immediately notify you and your loved ones that, oh, take you to the hospital just in case. And that can help like reduce heart attacks and deaths by heart attacks by tens of thousands every single year. At least in the US, heart attack is actually the leading cause, even above cancer. Like heart disease, sorry, not heart attack, is the leading cause of death, even above cancer. So these kind of devices would be very, very, very useful here. And even outside of, I guess, medicine, this can be used to clean oil spills. So you can target nanotechnology to selectively bind and destroy or disintegrate oil particles. So when you have oil spills in water, in oceans or reservoirs, which kill lots of innocent sea creatures or keystone species within the ocean, then you can use these kind of technologies to go and get rid of these oil particles and make sure that they don't kill thousands of innocent creatures within the ocean. And that'd be a very cool technology as well. So does anyone have any questions on that? That's for nanotechnology, and we just have one more thing remaining, and it'll be very quick, like 10 minutes. So any questions? Okay, cool. And if you have any, again, feel more than free to type it into the chat. We'll cover the critical thinking questions during our private, when we go into breakout rooms. But the last thing we're going to cover today is biosensors, chat-wise. Yeah, I'm glad you found it interesting. I'm glad. So the last thing we're going to cover is biosensors, and this is personally my favorite area of science because everything I talk to you about today, aside from some types of CRISPR, is reactive. If I get a certain kind of disease, then using nanotechnology to help cure it, that kind of stuff. If I get impacted by certain kinds of cancer, can I try to excise it out of my DNA? That can be both proactive or reactive, maybe. But for biosensors, it's completely diagnostic. So it helps to show you, do you have a certain disease or not? And it might sound, I'm just trying to give you a very basic understanding of it before I go into the nitty gritty. But you can think of it as a pregnancy test for diseases. So if I was using a biosensor for cancer, it'd be a pregnancy test for cancer, or a pregnancy test for HIV, or a pregnancy test for malaria. So these are really cool systems that operate on something called ELISA assays, and we'll get into that a little bit later. So all biosensors are doing is that they're testing the presence of a protein. So it indicates, do you have a certain condition? So the same way that a pregnancy test operates is that a pregnancy test tells you, are you pregnant or not? But they do it based on the presence of a protein called HCG, um, human or something CG. I'm kind of forgetting, the name is kind of escaping me right now. But HGH, human growth hormone. My bad. But yeah, essentially what they're doing is that they're seeing if you have a presence of this protein, which only happens in the body if you're pregnant. And if that, if the sensor detects that protein, then it can say, yeah, you have that protein, therefore you must be pregnant. So then the question is asked, well, what if I can switch and modify this the same way we were modifying CRISPR to study, to detect cancer, or modify cancer versus AIDS versus autism versus mental health? What if I could do that with biosensors? And the answer is yes, absolutely you can. So the first thing we need to understand at a very, very basic level is antibodies. So you all must know antibodies from immunology. Like when you get sick, I think everyone understands that, if you're sick, if you have the flu, there's antibodies in your body. And what they do is that they try to bind to these bacteria, they try to bind to these viruses, and they try to eliminate these threats. And these viruses and bacteria all have different kinds of proteins and RNA strands, and that's what your antibodies are binding to. So they're all targeted, very hyper-targeted to one specific kind of protein. And that's where I believe the eagle analogy comes in again. That they will only target one kind of protein. So this eagle is dead set on having a white rat instead of a black rat. So it would only eat this black rat, right? So it won't eat any of the, only this white rat. So it won't eat anything else. So that's just like antibodies. They're very selective, and they only bind to one specific kind of protein. So now, as I was kind of mentioning, some can fight diseases and harmful proteins, and those are called like T cells. So those are like the assassins, those attack pathogens. But others actually detect diseases. So those are like B cells, those are memory cells. So those can kind of store and remember and see, well, are we encountered with a certain kind of protein? And they bind to these kinds of proteins. So these are the kind of antibodies that we're trying to exploit with biosensors. And in the chat, really interesting. Yeah, I'm glad you learned this in school. This is, yeah, it's a very useful part of immunology. So essentially, this is how the whole system works. That you have a biosensor at the bottom, you have an antibody that's kind of on top of the biosensor, and then you have a protein that bonds to the antibody. So there's three components here. So when your protein bonds to the antibody, your biosensor records changes or electrical output that the antibody releases. So there are many kinds of biosensors. There are some sensors that lead to a color change reaction. So the way this happens is that there's a protein, you don't need to know it. It's called horseradish peroxidase, HRP. So what it does is that when an antibody binds to a protein, it changes color. So this is very easy to visually see, that if I have like a thin piece of paper lined with antibodies, and if all of a sudden there's a line on the paper that turns blue or that turns purple, then I can say, oh, the antibody must have bound to something. Because obviously, these proteins, these antibodies, they're so small. They're so hard to see. So I obviously wouldn't be able to know if my antibody binded to a protein because it's so tiny. But if I could have all the antibodies all change color and have a color change reaction, then visually I can see that, oh, if my paper turned blue, then it's easy to see that then the antibodies must have bound to something. So that's a very interesting kind of process there. That's where the pregnancy test comes in. So over here, you see that there's one kind of line that shows you're not pregnant, and this second line appears if you are pregnant. So this is operating on this antibody conjugated to horseradish peroxidase, conjugated to an enzyme that changes color if it binds to a protein. So over here, when you're taking the pregnancy test, if your urine has, I believe it's here, HCG, if your urine has this kind of protein, then what happens is that, the antibody and the horseradish peroxidase has a color change reaction, and you can see that color change reaction very visually. So that's this extra pink line. If this was urine from, let's say, anyone like a man that's not pregnant or whatever, obviously, then this would be completely white because there's no protein present there, so you don't bind to anything. And if you're not binding to anything, then there's no color change reaction because there's nothing to see. And yeah, that's how it works in a pregnancy test. So then the whole question that occurs is that, well, why can't I switch this antibody out to detect another kind of protein? And this goes all again with the dragon analogy. And I told you to come back, right? That if I change my dragon's crystal from white to red, it goes from light dragon to fire dragon. If I change it to green, it goes to like a grass sky type dragon. If I change it to purple, then it goes to like a dark type dragon. So as I change the color of my crystal, the type of dragon I have changes. Likewise, if I change the type of antibody I'm using, so I could use an antibody for the HCG protein, the pregnancy protein, and I would have a biosensor for pregnancy. But what if I use an antibody that's for a CA125 protein? So I could apply the same concept to cancer. I'll go over this a little bit. I'll first show you the visual and I'll go over this. So don't worry about that. But yeah, what if I change my antibody to be specialized to CA125? And CA125, it's called cancer antigen 125. This is a cancer protein for ovarian cancer. So it's very cool because it's a protein that's only expressed on ovarian cancer cells and not expressed on healthy ovarian cells. So if anyone's parents has ever had ovarian cancer, or your grandparents has had ovarian cancer, you know that when they take, if they go to the hospital, they take blood tests. And when they take blood tests, they tell you what your CA125 level is based on how much of this protein you have in your blood. So there's obviously so many complications to this because you need to draw blood from them. That's painful. Oftentimes they might be immunocompromised, or they might not be able to heal as fast. So that's a painful process. They have to go to the hospital multiple times. And especially in the time of the pandemic, that can be really, really risky because, you can contract the COVID virus. So there's all these hurdles and all this painstaking process. But what if I told you that, in just one drop of blood, or maybe even a drop of saliva, that I could go and see if there's CA125 cancer proteins in your body, and then I can immediately tell you whether you have ovarian cancer or not. That's a very, very useful thing to do, and that's why a lot of work is being done here. And some of you may be wondering, well, how do you know if it's not a color change reaction? How else would you be able to do it? You can actually do it through an electrical reaction. So I'll be covering this with the advanced group again, but if you had your antibody conjugated to it's called a single-walled nanotube, a single-walled carbon nanotube. Essentially, what would happen is that instead of a color change, when the antibody binds with the cancer protein, what happens is that you see an electrical signal be released. So then the biosensor can go and try to detect, is there an electrical voltage differential? Like, is there a difference in the electrical signal? And if it is, you can have it light up. You can have it make a noise. You can have it make a ding noise. You can have it light up. You can have it do anything you want. And that's all with the programming end. But that makes it even easier because I don't have to look for, is there a color change? My sensor will beep, or my sensor will just light up, or my sensor will send an alert to my smartphone and tell me if I have cancer or not. So this is something I wanted to make everyone aware of, because just like CRISPR, we can apply this to any kind of disease that we want. So we can literally go and say that, are we trying to detect if you have, let's say, autism? And we narrowed autism down to specific kinds of proteins only responsible for autism. That's really hard to do. But if we could, imagine if we could imagine that we could, then we could detect, does your child have autism? Just as simple as that. If you wanted to say, do you have malaria? Is this a normal sickness? Or a big problem that a lot of people are facing during the COVID pandemic is that, well, if I'm sick right now, if I just caught a cold because I wasn't sleeping enough, or I didn't wash my hands before I ate or something, do I have like this life-threatening coronavirus illness, or am I just sick? What we could do is that we could modify an antibody specific to a COVID protein. And we have slides on this for everyone to look through throughout the throughout our breakout rooms. But if we wanted to modify the proteins, modify this antibody selective to COVID proteins, then just as easy as putting a little bit of spit, like just putting some of your saliva on this biosensor, you'd be able to detect, do you have coronavirus or is this just a normal sickness? And you don't have to worry about quarantining and not seeing your loved ones. You don't have to worry about going to the hospital and risking contracting coronavirus. So this would be a major, major helpful thing. And this could apply for any disease any of you are passionate about, be it malaria, be it AIDS, be it Huntington, be it anything. So it's a very kind of cool technology. So with that, does anyone have any questions on biosensors? Because it's definitely a little complicated. Or just anything in general, if you have any questions on anything in general. Okay, that looks good so far. So what I kind of wanted everyone to do is is kind of look at these critical thinking questions. And while we're kind of working with the instructors to sort you all into your private groups and everything, just answer these questions either in your head or maybe just on maybe just in front of you on pen and paper or whatever. And we'll be talking about this as kind of like a collective class. But just take 3 minutes. I'm just gonna work with the instructors to sort you into your private groups. But just take 3 minutes to just read these questions over and kind of think about them on your own. So again, just think about what other diseases and conditions we could use biosensors to detect other than the ones I covered, like cancer. What happens if I place my biosensor somewhere very cold or very hot? How could that affect the way it functions? Because that's very interesting to think about how they work. And lastly, how could these change the way medical disease detection works in hospitals and at home, for elderly patients, for children, just all in general? So just consider these 3 questions and I will be with you all shortly. So just in 3 minutes. Okay, so this is one application that I wanted to go over with everyone for CRISPR. So this, some of you may or may not know, but COVID-19, is coronavirus, and H1N1 was like the swine flu. So it was another kind of big, almost like pandemic kind of virus, and they both replicated through a similar way. So let me kind of use this pointer function here to help illustrate what's going on. So over here, we have a virus, and a virus has its viral DNA, viral genetic material, I should say. It has RNA because it's a retrovirus and has proteins on its surface. So kind of similar to cancer cells, they have these unique kinds of surface proteins that they express on their coat, and then they have a certain kind of DNA sequence that they, RNA sequence, a genetic sequence that they encapsulate. So when they enter into a cell, what happens is that these viral particles actually release their RNA. And this is a more intermediate kinds of steps that we'll cover in next week's chapter of how viral replication happens and how genetic division happens. But essentially what happens is that they translate their RNA and basically divide it multiple times. So they replicate it, they have it grow multiple times. Then they integrate it into the cells, into the normal cells, like your, let's say, lung cells DNA. And then they have your cell copy that genetic material again and again and again, and that's how they grow. So the interesting thing about viruses is that they're not technically alive. So they rely on you to be alive to replicate the virus. So viruses cannot divide on their own. The same way that our cells divide on their own, our cells copy the genetic material, divide, reproduce, replicate. Viruses have to integrate themselves into our cells' DNA so they can reproduce as well. So they rely on us to help them reproduce. So what's interesting is that the viral RNA, the viral genetic material is very, very bad. And this is what allows for the virus to be made. So when this integrates into our genome and we help the virus replicate, then billions of new viruses are created within our bodies by our own cells that basically go and fill up our cells like they're a balloon. So I always give this analogy that imagine I have a balloon and imagine I blow in it once, right? That's a normal size balloon. That's a tiny balloon, nothing. Imagine I blow in it 10 times. Okay, it starts to be a big balloon. But imagine if I blew into it 1,000,000 times, right? At some point, the balloon would get big, 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 and then it'd pop. So this is a very similar mechanism to the way viruses work in cells, that they replicate. And initially, it's fine for the cell. It's like, whatever, they're tiny virus particles. But then when there's millions of them inside one tiny cell, and the cell just bursts open, it lyses. And that's exactly what viruses do. So that's why when you are sick, you get a sore throat, because a virus is invading a lot of your throat cells. And as they're popping your throat cells, you're getting pain reception. So that's why your throat is hurting. So then the viruses, as the cell pops, the millions of viruses spill out everywhere throughout your bloodstream, throughout your body, and they infect everywhere else. So that's exactly what the COVID virus does. So what CRISPR can do essentially is say that, well, why can't we look at these viral RNA strands and target these RNA strands specifically? Now, an interesting thing about CRISPR is that it doesn't just have to target DNA. CRISPR can target RNA, it can target mRNA, it can target proteins, it can target rRNA. So CRISPR is very, very, versatile. So we can target any kinds of genetic material. So why not COVID? Why not coronavirus? So what a lot of people have been trying to do is, this is like a research headline, but they've been trying to use the CRISPR system to basically inhibit the way that the coronavirus replicates in our body. So there's a lot of complex diagrams and everything here, but the takeaway here is basically when your virus is trying to integrate itself into your genome and trying to express its viral genes, what we can do with CRISPR is we can attack the viral RNA there and prevent it from ever being packaged or released. So even if, you know, the virus tries to infect your cell, it can't do anything, right? It can't even harm you because there's no way for it to package and assemble into a virus. So all it is just a bunch of, you know, parts that are laying down. Like another way to think about it is like, you know, if there was a scary killer robot, I give this example all the time, but if there's a scary killer robot, you know, that's a very scary killer robot. You wouldn't want to be near that. That's a threat to your life. But if I just gave you if I disassembled the robot and I showed you its engine and I showed you its circuitry and I showed you its robotic arm and I showed you, you know, some mechanical part, you'd be like, oh yeah, that's boring. That's just, you know, that's nothing. So that's basically what we're doing to the virus. We're crippling the virus through that way. Does that make sense to everybody? Okay. How does coronavirus replicate? Yes, very good question. So it is a retrovirus. So it does use reverse transcriptase like HIV. So it inserts its RNA into the cell. And what happens is that it uses reverse transcriptase to make that into cDNA, complementary DNA, integrate that into your DNA, right? Because we don't have RNA, we have DNA. And then it has that be replicated by your own cell. So all we're trying to do is catch the RNA before it can go and integrate itself into your DNA as cDNA. And then it's never able to integrate, it's never able to package, it's never able to harm you. And another application of CRISPR, someone actually brought up, I think it might have been Miriam, I'm not sure, but essentially someone brought up the use of, brought up the use of CRISPR for malaria. And that's a very interesting kind of application. So there has been a study published, and this is just ahead of the study, but basically it's like, I'll just read it out for you. A CRISPR-Cas9 gene drive targeting double sex causes complete population suppression in caged certain Anopheles gambiae mosquitoes. So what this means is that basically they're trying to look at the way that we can suppress the gene that causes mosquitoes to divide, to mate, and to have offspring for malaria. So what this is essentially doing is that they're saying that, well, if malaria is transmitted through mosquitoes, if we can modify mosquitoes to never even bite us in the first place, to never even be able to have kids in the first place, then we won't have malaria. And that's a very, very cool thing to do. So that's why that's where this kind of comes from. So there's this whole phenomenon called genetically modified flies, GM flies, where we can essentially modify mosquitoes so they're sterile, and when they mate with other mosquitoes, they don't give birth to any children. So mosquitoes they divide, they have kids, mate rapidly, right? One mosquito can give birth to hundreds of other mosquitoes, and that can be very, very harmful. But what we can do here is that we can essentially put a few modified mosquitoes out into the population, and these mosquitoes will basically outcompete other mosquitoes. They'll mate with all the mosquitoes out there, and then the mosquitoes will have no kids because these mosquitoes will be sterile. So then all the mosquitoes will die out, and that could be a good thing because then there's less malaria transmission because there's less mosquitoes transmitting malaria. It could be dangerous because if there's less mosquitoes, then would that impact other species? Would that impact frogs that rely on mosquitoes to live, that rely on other kinds of animals that, prey on mosquitoes? And the answer is kind of complicated. So the answer is yes and no. There aren't too many species that only rely on these kind of mosquitoes to divide and to live and sustain themselves, but there are definitely some that rely on the mosquitoes. So it's a win and lose situation where we might save a lot of people from dying from malaria, but we also might kill a lot of animals that might have other negative downstream effects. That's something we need to be aware of. And now past that, here is another kind of application of CRISPR in agriculture. So this is another study head that talks about rapid improvement of domestication traits in an orphan crop by genome editing. Now that sounds very, very complicated, but to break that up into smaller parts, all that's doing is that it's basically making it so that orphan crops, these are kind of standalone crops that are different from their genetic parents. How can we modify them to grow better? How can we modify them to grow without seeds, or grow more efficiently, or grow in soil that's less ideal? So grow in soil that's more dry, that has less nutrients. And how can we modify them such that they are good for crop yield? And this, I'm kind of using this as a starting point to talk about the way that we can use CRISPR for plants. So CRISPR is just modifying DNA, right? It can be mice DNA, human DNA, and even plant DNA. So if I can modify a plant's DNA so it can grow in really hot temperatures with very little water, that'd be an amazing thing. So I'm sure I don't know if any of you are from more rural regions, but I've had students in rural regions talk to me about this, about how a lot of their water goes to waste when they water their crops. And it goes to, if you have crop rot, then all the water you wasted on growing that crop goes to waste. If it's a hot day, then and it's dry air and not humid, then you might have evaporation. So your water might dry out and evaporate before it even reaches the plants. So what if we could modify plants so they barely need any water? And there are specific genetic sequences in plants which control how much water they need, which control how they grow and everything. So if you can modify that, that'd be amazing. Or certain kinds of plants, I'm sure you all might know this, like there's different kinds of nutrients in soil, right? So some soil might be good for corn, but might be bad for barley. Other soil might be really good for growing like an apple tree, but might be terrible for growing like eggplants. So it really varies a lot. So what if we could what if I had one specific kind of soil that was bad, and I can modify my plants so they would be happy with whatever nutrients are in the soil? There's ways to do that and edit the plant genome to do that. And that's a really interesting question of, would that food be healthy? Would that food not lead to like cancer if I was to consume it? Would that be edible? All these kinds of different questions. And that's what scientists are trying to parse out right now. Well, yeah, I'm glad. I'm glad everyone kind of was on top of everything, and I'm glad you all enjoyed it. And I'll see you all next week, same time on Wednesday.